I'm very happy to present our latest work, mEnrich-seq methylation-guided enrichment sequencing of bacteria taxa of interest for microbiome. So some background to start with. So our human body is a very rich resource for all kinds of microbiome, and a metagenomic sequencing is a great tool to profiling all the genomes in one given sample. But we do see the increasing needs for sequencing some specific bacteria, like some pathogens, the pathogenic E. coli from the urine of urinary tract infection patients, or we want to know some beneficial bacteria like the bifido bacteria strains in different people from the gut microbiome sample. For all these bacteria microbiome samples overwhelmed with human DNA, we do have a solution, like using the saponin or the host DNA depletion kits to remove the host DNA. But none of the current methods can effectively differentiate bacteria species from each other. So we're thinking whether we can come out with a strategy to enrich a specific microbiome, yet to differentiate it from others. So we take a step back to look at the epigenetic feature of the bacteria. The DNA methylation is ubiquitous in bacteria because it is a very important part of the restriction-modification system, which helps the bacteria to defend against the invading DNAs. And this RM system can be divided into 3 different types, generating vast variety of methylation motifs here. So speaking of the methylation motifs, the bacterial epigenome is highly motif-driven. Only a very few motifs in one bacteria are methylated. And for one given methylation motif, over 95% of its sites are methylated. So one of our previous lab member, John, has already taken use of this DNA methylation information as a natural epigenetic barcodes to do the methylation binding after sequencing. So with this DNA methylation information can help resolve the assembled contigs and individual reads into species and even strain levels. So we ask a question, this is before sequencing. Can we do something like take use of this methylation information right before sequencing the whole microbiome sample? So leads us to the core idea of mEnrich-seq. For a given microbiome sample here, the bacteria that shares the same methylation patterns are circled in the one color here. And some bacteria may share multiple methylation patterns, so it can be covered by 2 different colors here. So the scissors here represents all the restriction enzymes. When using the right restriction enzyme to the microbiome sample, the background DNA can be largely depleted while leaving the bacteria in the red circle intact. So with this core idea, we incorporate it with our library preparation steps with the highlights of the gel purification to largely get rid of the background DNA, and also the PCR amplification, which help us to enrich our target species. And more importantly, our current method is generally compatible to all sequencing platforms. So for next few slides, I will give some real applications. Firstly, we take use of mEnrich-seq to enrich the pathogenic E. coli from the urine sample, which causing the urinary tract infection for these patients. This is a scatter plot. The x-axis stands for the relative abundance of the species in the original sample, while the y-axis represents the relative abundance after using the mEnrich-seq. So with mEnrich-seq, we can significantly enrich the E. coli as high as 116-fold and reaching 90% of relative abundance. So in addition to the fold enrichment, we are also caring about the completeness of the E. coli genome. So based on our analysis, the reads from the mEnrich-seq can cover both the chromosome and plasmids of the E. coli without systematic bias and through all 3 samples. So next, I will show the example trying to use the mEnrich-seq to enrich a beneficial bacteria Akkermansia from the infant fecal sample. We are focusing on this bacteria because it's gaining more and more interesting due to its association with many human diseases and linked with patients' responses to cancer immunotherapies. And this bacteria is still relatively hard to culture. So with mEnrich-seq, we can reach a very great fold enrichment for the Akkermansia and together with some other interesting bacteria. And similarly, the reads come from the mEnrich-seq covers the genome of Akkermansia without systematically bias as well. So the previous 2 cases demonstrate the use of mEnrich-seq for enrichment of some bacteria with known methylation motifs. So what happened if we don't know the methylation motifs yet? Here we'll show the example trying to enrich the bacteria with de novo discovered methylation motif. Here is a very simple workflow. For a given metagenomic sample, we are firstly doing an initial sequencing. And for those bacteria with relatively low abundance cannot have a very good assembly context, we're using the existing tool to call the methylation motifs de novo, and then select the restriction enzymes rationally according to the motifs discovered. And coupled with these REs, we will use mEnrich-seq to enrich the bacterial texture of interest. So for demonstration purpose, we here will demonstrate the use of either individual restriction enzymes or multiple REs in combination to enrich bacteria from an adult fecal sample. So when using the single or individual RE, we can already significantly enrich 6 bacteria species, here as high as 43-fold. And paying attention to this bacteria, it harbors 2 different methylation motifs and can be targeted by multiple REs. So when using the multiple REs in combination, we can significantly increase the relative abundance to 60% and also as high as 85-fold enrichment. So here's a very good example. We are targeting some bacteria of interest based on the de novo discovered methylation motifs. So at this point, people will always ask, is that all the bacteria you can target, or how many more we can target by using the mEnrich-seq? So we did a meta-analysis of the methylome of over 4,000 bacterial strains, and over 60% of these strains have at least the methylation motif that can be targeted by mEnrich-seq when coupled with commercial available RE. And then we build a phylogenetic tree here. So all these strains that can be targeted by mEnrich-seq demonstrate a very broad taxonomy diversity. So the conclusion here is that mEnrich-seq shows a broad applicability for bacterial enrichment. So last slide will be the take-home messages. So here we demonstrate our new method, mEnrich-seq, which can enrich a bacteria of interest effectively at very high completeness for their genomes. And we can also enrich some bacteria with unknown methylation pattern by de novo motif discovery approach. And most importantly, mEnrich-seq is broadly applicable. So I will take this chance to thank my supervisor, Professor Gang Fang, for his full support for all these years and this big project. And also I will give my thanks to all the lab members and the collaborators from Mount Sinai and Reuters University. So that's it. Thank you.
Thank you.